During the American Legion Convention in Philadelphia in 1976, 221 people contracted pneumonia, and 34 of them died from an unknown ailment. The agent responsible for this outbreak was then identified as Legionella pneumophila, marking its place as the first named member of the Legionellaceae family. About 20 species of Legionella have been isolated from humans. They are ubiquitous gram-negative bacilli acquired through inhalation. Legionella infection can exhibit a range of symptoms from no visible signs to severe illness. To diagnose the infection in a laboratory setting, any one or a combination of the methods below can be used: isolation using special media, detection of urine antigen, direct fluorescent antibody, serology. Most human cases of legionellosis are caused by Legionella pneumophila. Other important species are Legionella mcdedii, Legionella longbeachiae, and Legionella dumoffi. Several factors can influence the various presentations of Legionella species infection. These may include the organism's ability to invade and survive within host cells, particularly bronchoalveolar macrophages, as well as its capacity to generate proteolytic enzymes. While Legionella species are known to cause intracellular infections in humans, they are also capable of thriving in an extracellular environment. Legionella infections can manifest in several ways: febrile disease with pneumonia, called Legionnaire's disease; febrile disease without pulmonary involvement, called pontiac fever; and asymptomatic infection. Notably, the transmission method and number of infecting organisms in the inoculum are influential factors in the infection's clinical features. Moreover, individual susceptibility to Legionnaire's disease is heightened by several host factors, including a suppressed immune system, chronic lung disease, alcoholism, and heavy smoking. Manifestations of Legionnaire's disease can be classified into three main patterns: sporadic cases, which occur most commonly in the community; epidemic outbreaks, which are characterized by low attack rates and short duration; nosocomial clusters, which occur in populations of compromised patients. Legionnaire's disease caused by L. pneumophila has several serogroups, with serogroup 1 accounting for approximately 85% of cases. In addition to serogroup 1, other frequently implicated serogroups are L. pneumophila serogroups 4 and 6, L. longbeachiae, and L. mcdedii. The incubation period for Legionnaire's disease is 2 to 10 days, with patients initially experiencing a non-productive cough, fever, headache, and myalgia. As the disease progresses, pulmonary infiltrates may develop, resulting in bloody or purulent sputum. Other clinical manifestations include rales, dyspnea, and shaking chills. Pontiac fever is a non-pneumonic form of legionellosis. The incubation period is brief, approximately 2 days. Patients are typically healthy individuals who experience flu-like symptoms, including fever, headache, and myalgia. These symptoms usually last for 2 to 5 days and resolve on their own, without the need for medical intervention. Legionella species has the ability to colonize a variety of sources. The following are contributing factors that aid in their colonization: multiplying effectively between 20 degrees Celsius and 43 degrees Celsius, and surviving for varying periods between 40 degrees Celsius and 60 degrees Celsius, adhering to pipes, rubber, plastics, and sediment, and persisting in piped water systems even after flushing, surviving and multiplying within free-living protozoa and in the presence of commensal bacteria and algae. Several methods, such as direct examination, culture, and antigen and antibody detection, are available for the laboratory diagnosis of infections caused by Legionella species. Legionnaire's disease is best diagnosed using a combination of culture and urine antigen detection. Diagnosis of Pontiac fever is usually limited to serology. Most laboratories use more than one method to maximize their diagnostic capabilities. Collecting and processing specimens for Legionella infection diagnosis. When testing for Legionella infection, specimens for culture and direct examination typically include sputum, bronchoalveolar lavage, and bronchial washings. Other tissues or fluids may be submitted in sterile, leak-proof containers when suspicion is high. If submitting small pieces of tissue, overlay them with sterile water and avoid using saline or buffer, as they can inhibit Legionella species growth due to the presence of sodium. To prevent overgrowth of contaminating microbiota, refrigerate specimens if delays of over 2 hours between collection and processing are unavoidable. If processing will be delayed for several days, freeze specimens at -70 degrees Celsius. Urine is an important specimen to be collected for antigen detection, but it is most useful for L. pneumophila serogroup 1. Specimens are collected in sterile, leak-proof containers and should be stored at 2 degrees Celsius to 8 degrees Celsius or frozen at -20 degrees Celsius if testing is delayed. Environmental sources may be cultured during epidemiologic investigations, but this is normally a public health rather than a clinical or diagnostic function. Under a microscope, Legionella species appear as gram-negative bacilli that are pleomorphic and weakly staining, measuring about 1 to 2 micrometers by 0.5 micrometers in size. To enhance the staining intensity of the organisms, extend the safranin counterstaining time to a minimum of 10 minutes. L-mictadie is weakly acid-fast in tissue and is best stained using the modified Kinyoun procedure. For better observation of the organisms, other stains such as Diffquick and Gimza can also be used. If you suspect the presence of L-mictadie, look for faint staining, pleomorphic gram-negative bacilli outside and inside macrophages and segmented neutrophils. The modified Kinyoun procedure for tissue testing can be used for this purpose. Isolation techniques. When testing for Legionnaire's disease, it is essential to culture the organism. Even when utilizing a rapid test, such as urine antigen detection, this should always be attempted. To isolate Legionella species from specimens contaminated with other bacteria, such as sputum, acid treatment can be applied to enhance the process. The medium is then inoculated with a portion of the treated specimen, which is then incubated at 35 degrees Celsius to 37 degrees Celsius in air for at least 7 days. Legionella species colonies will typically emerge within 3 to 5 days. Isolating Legionella species. Legionella species are fastidious, aerobic bacteria that require L-cysteine for growth and do not grow on sheep blood agar. Although tiny colonies may appear on chalk agar, the best option for Legionella isolation is buffered charcoal yeast extract BCYE agar with L-cysteine. Fortunately, BCYE agar is commercially available as a non-selective and semi-selective media, both of which should be used for optimal isolation. Semi-selective BCYE agar contains polymyxin B and isomycin and either vancomycin or cefamandole, which can improve recovery of Legionella species from highly contaminated specimens. However, it is important to note that semi-selective medium can inhibit growth of some Legionella species, and so it should not be used alone. BCYE agar colonies have distinct visual characteristics. They appear as convex, glistening, and grayish-white or blue-green in color, with a diameter of 2 to 4 millimeters. Young colonies have a granular, light gray ground glass center and furrowed, pink, light blue, or bottle green bands on the periphery. It's essential to inspect the plates daily, as older colonies lose their unique features and may be confused with other bacteria. To distinguish autofluorescing colonies from non-autofluorescing colonies, suspicious plates can be illuminated with a long-wave ultraviolet light, 366 nanometers. Identifying Legionella genus colonies. When identifying suspected Legionella colonies, it is possible to use a presumptive method to determine their genus. This method involves testing for growth in L-cysteine, performing a gram stain, and using direct fluorescent antibody tests. Although biochemical testing has limited value in the further identification of isolates to the species level, following this protocol can be helpful in evaluating suspected colonies. Here are the steps involved. Gram stain any suspicious colony growing on BCYE agar. Look for thin gram-negative bacilli. Legionella spp may vary in size from 2 to 20 meters in length. Subculture the isolate to BCYE agar with L-cysteine and to either sheep blood agar or BCYE agar without L-cysteine. Legionella species thrive on BCYE agar that has been enriched with L-cysteine. Smear preparations should be made from colonies that require L-cysteine and tested with polyvalent and monovalent conjugates to determine specific species and serogroups. If definitive identification of less common species is required, it is usually performed at a reference or public health laboratory, often using 16S ribosomal RNA sequencing methodology. Rapid techniques for Legionella infection detection. Urine antigen test, a highly specific method with a 97.1% sensitivity to detect the Legionella antigen in patients. Keep in mind that this test may not be as effective for individuals with recent infections or those who have had the infection for an extended period of time. The direct fluorescent antibody test, DFA for short. The DFA test is a quick, laboratory-based procedure used to identify the most common Legionella strains present in clinical samples obtained from the lower respiratory tract. Here's how it works. The conjugate in the test binds to antigens on the cell surface. Next, a fluorescence microscope is used to detect the antigen-antibody complexes. When viewed under a microscope, Legionella species bacteria appear as short or coccobacillary bacilli with vivid peripheral staining. They can range in color from bright yellow to green. It's crucial for microscopists to have experience working with the morphologic and staining properties of Legionella spp so that atypical organisms are not mistaken for Legionella. Although the DFA test is highly specific, 94% to 99%, some strains of Pseudomonas, Bacteroides, Corynebacterium, and other bacteria can cross-react with the polyvalent conjugates. Some labs opt not to use polyvalent reagents for direct specimen examination due to these cross-reactions. DNA detection methods. Various DNA detection systems are available for laboratory use. Although nucleic acid amplification tests like PCR and strand displacement amplification show promise, they are not yet commonly available for routine clinical testing. Thank you for tuning in. If you found our video to be informative, please show your support by liking and subscribing to our channel.